We always talk about the brain as this supercomputer. We get obsessed with the neurons, the processing chips, the firing rates, but we almost never talk about the cables.
Which is funny, right? Because without the cables, that supercomputer is just a useless lump of gray jelly.
Exactly. And today, we're doing a deep dive into the insulation around those cables. It's a story that starts with a tiny molecular sensor, and believe it or not, it ends with how human babies learn to walk. And frankly, it completely upended how I thought biology worked.
Oh, it's a huge paradigm shift. We're looking at a paper published in Neuron by Duretti and their team, just out in early 2026, and they've basically solved a mystery that has been bugging neuroscientists for literal decades.
Okay, before we get to the solution, you have to lay out the mystery for me, because I thought we kind of knew how this worked. You have a nerve cell, it has a long tail called an axon, and you wrap it in fat called myelin to make the signal go fast. That's, textbook stuff.
That's the what. The mystery is the how. Specifically, the sheer logistics of it. The cell that makes the myelin in your brain is called an oligodendrocyte. Let's just call it an olig for short.
An olig. Got it.
Now, I want you to picture an octopus. But instead of 8 arms, this thing has up to 60.
60.
Up to 60. And it's reaching out in 3-dimensional space, trying to grab onto 60 different axons at the same time to wrap them all up.
60 different wires at once. That sounds like an absolute multitasking nightmare.
It gets worse. Those 60 wires are all different sizes. Some are thick, high-speed data cables. Others are tiny little threads. Now, if you're that octopus, how do you know how much insulation to put on each one? You can't just use a standard roll of tape. The big cables need 40 layers. The tiny ones might not need any.
Okay, now I see the problem. I always just assumed there was some kind of, chemical conversation happening. Like the big axon shouts, "Hey, I'm massive over here, send more fat."
That is a perfectly logical assumption. And in fact, that's exactly how it works in your arms and your legs. In what we call the peripheral nervous system, the nerves release a protein called neuregulin-1. It's a chemical signal. The more signal, the bigger the nerve, the more wrapping it gets.
So scientists just figured the brain did the same thing.
For years. They hunted and hunted for that chemical signal in the brain. They looked for neuregulin, they looked for other proteins. But here's the big twist. The oligodendrocytes in the brain basically ignore those chemical cues.
They don't care about them.
They don't care about the chemical conversation at all.
So the brain is wrapping 60 wires of different sizes, completely in the dark, without talking to them. That sounds like a recipe for disaster. How does it know what it's even holding?
It feels them.
It feels them?
Literally.
Oh.
Physically. The huge breakthrough in this paper is that the mechanism isn't chemical at all. It's mechanical. The cell is using physics to measure the size of the wire.
Wait, hold on. You're telling me a squishy biological cell is doing?
Yep.
What, geometry?
In a way, yes. Think about it. If you wrap a sheet of plastic around a really thick pipe, you have to stretch that plastic a lot more than if you wrap it around a thin little straw. The membrane is under physical tension.
Okay, that makes sense physically. But how does a cell know it's being stretched? It doesn't have a brain of its own.
It has a sensor. And this right here is the star of today's show. The researchers identified a very specific protein called TMEM63A.
TMEM63A. Okay, catchy name.
I know, rolls right off the tongue. But I want you to think of TMEM63A as a strain gauge. You know those little sensors engineers put on bridges to tell if the steel is bending too much?
Yeah, if the bridge flexes, the sensor trips and sends an alarm.
That's exactly what this protein does. It sits there in the cell membrane, the skin of the oligodendrocyte. When a membrane stretches, because it's trying to wrap a big fat axon, that physical tension literally pulls this channel open.
It pulls the door open.
Physical force opens the molecular gate. And it's what's called a high-threshold mechanosensor. That means it ignores all the little jitters and vibrations of daily life. It only opens when there's a real significant stretch.
Okay, so the door is open. What comes flooding in?
Calcium.
Of course. It's always calcium in these stories, isn't it?
It usually is. But there's a nuance here that's really, really cool. Usually when a cell uses a calcium signal, it releases it from its own internal storage closets, places like lysosomes. It's like getting a snack from your own pantry.
Right.
But the researchers proved that TM63A lets calcium rush in from the outside. It opens the front door and lets the whole neighborhood in.
How on earth could they tell the difference? A calcium atom is a calcium atom. You can't ask it where it came from.
They use zebrafish. These little larvae are completely transparent, so you can literally look right into their living brains with a microscope while they're developing.
Wow.
And they genetically modify the fish so the myelin would flash, literally light up, anytime there was a spike in calcium.
That sounds incredible to see.
It is. In the normal, healthy fish, they saw these beautiful, constant little sparks right at the leading edge of the myelin sheath, like tiny welding sparks as the cell built the wrapper.
And in the mutants?
The ones where they deleted the TM63A sensor, it was dark. Almost no sparks. The membrane was still stretching, but the signal just never happened.
Okay, let me pause you there because this is a huge pivot. I always pictured cells sort of tasting the water for chemicals, but you're saying this is purely tactile. The cell is essentially blindfolded, and its only instruction to build is the feeling of its own skin getting tight.
That is the perfect way to visualize it. It's the tactile theory of myelination. You don't need a chemical permission slip. You just need pure mechanics.
So just to recap this first part, the big problem is how a brain cell, an olig, knows how to wrap dozens of different sized nerve fibers correctly.
Exactly, without the chemical signals that are used everywhere else in the body.
And the answer is this protein, TMM63A. It's not a chemical receptor, it's a physical stretch sensor.
Right, a strain gauge.
When it feels the stretch of wrapping a big axon, it pops open, lets calcium flood in from the outside, and that creates a signal, a little spark that says, build here.
You've got it. The cell is literally feeling its way to a solution.
So if that sensor is gone, if you delete that gene, is the cell just numb?
It loses its sense of touch, and this is where we get into what I call the Goldilocks failure.
Goldilocks, as in too hot, too cold.
Exactly. The researchers created a mouse model where they deleted the TMM63A gene, the knockout mouse, and they looked at what happened to the brain's wiring. Remember, a healthy cell is picky. It ignores the tiny wires and wraps the big ones just right.
Right, custom tailoring for each axon.
But without the sensor, the cell gets completely confused. It starts applying a kind of one-size-fits-all approach to everything. First, looking at the big axons, the ones that need thick coats, they were underwrapped. That's hypomyelination. The insulation was way too thin.
So the heavy-duty cables are left exposed. That can't be good.
Not at all. But then the really strange part is when they looked at the tiny little axons, the ones that are normally supposed to be left naked.
Yeah.
The confused cells started wrapping them too. They found what they call ectopic myelin on wires that were way, way too small to need it.
So it's just throwing insulation at everything randomly, like a mailman just stuffing letters into any mailbox he sees?
Precisely. It's blind wrapping. And this messes up the fundamental geometry of the brain in a really specific way. The paper talks about the internode and the node of Ranvier.
Let's define those fast. The internode is the insulated sausage, and the node is the little gap between the sausages.
Perfect analogy. The internode is the insulated segment. In these blind mice, the internodes were, on average, 22% shorter.
22%? That's a lot. So short sausages.
Which means you need more links between them. The nodes of Ranvier, those gaps where the signal has to jump, became more frequent, and the gaps themselves were physically wider.
Does the width of that tiny gap really matter that much?
Oh, it matters immensely. Think of the nerve impulse like a sprinter who's jumping over hurdles. If the hurdles are nice and tight and evenly spaced, the sprinter gets into a rhythm and goes fast.
Right.
But if the hurdles suddenly become huge, wide mud pits, the sprinter slows down, stumbles, and loses their rhythm. The signal gets sluggish.
Sluggish.
It's actually worse than sluggish. It gets out of sync. They measured the electrical signals in these mice. The main signal, the fast one, was slower and weaker, as you'd expect. But the slower signals showed something called temporal dispersion.
Temporal dispersion sounds like a glitch in a time travel movie.
It kind of is for a nerve signal. Think of it like an orchestra. If the violin section plays perfectly in time, the sound is crisp and powerful. Temporal dispersion is like half the violinist coming in a split second late. The sound is muddy. The signal arrives, but it's smeared out over time.
And what does a smeared signal actually look like in a living mouse? Is the mouse dumb? Is it paralyzed?
Not dumb and not paralyzed. Clumsy. They put these mice on a rotorod, which is basically a spinning log, and they analyzed their footprints with a catwalk test. The mice could move, but they had what they called fine motor deficits. Their gait was uneven. They tripped more often. Their coordination was just off.
Because the timing signal in their brain was muddy.
Exactly. High-performance movement like catching a ball or for a mouse running on a spinning log, it requires millisecond precision.
Yeah.
If your myelin is generic and off the rack, instead of being custom-tailored, you lose that precision.
Okay, let me do a quick recap here because this is fascinating. Without the TMM63A sensor, the cell becomes indiscriminate.
Yep, it's blind.
So large axons get underwrapped, meaning their insulation is too thin and the segments are too short.
Right.
And tiny axons, which should be bare, they get accidentally wrapped. This makes the nodes of Ranvier, the gaps, too wide, which slows down and muddies the nerve signal.
Creating that temporal dispersion.
And the end result isn't paralysis, but a loss of coordination. Clumsiness.
You nailed it. It's a brain that works, but it's not a high-performance brain. Which raises the next big question. We have a calcium spark at the beginning, and we have a poorly built wall of myelin at the end. What happens in between? How does a burst of calcium actually tell the cell to start building?
Right, this is the supply chain part of the story.
Exactly. So the researchers did this massive census, a transcriptomic analysis of all the genes that were active in these mutant mice. They wanted to see what exactly was broken. And they found that most things were actually fine, but one specific department was failing badly. Vesicular transport.
Which is trucking.
Molecular trucking. And they zeroed in on one specific protein called MYO5A. Myosin 5A.
I feel like I remember myosin from high school biology. It's in our muscles, isn't it?
Same family, but a different job. In the brain, MYO5A is a motor protein. I want you to visualize a tiny two-legged creature walking along a tightrope inside the cell. That tightrope is the cell's cytoskeleton.
It's carrying something.
It's carrying a backpack. And inside that backpack are the blueprints for building myelin. Blueprints. Not the bricks themselves.
And this is a fascinatingly efficient quirk of these cells. They don't make the myelin protein in the center of the cell and then ship the finished product out. That would be like baking all your bricks at headquarters and then driving them to 100 different construction sites. It's too heavy and way too slow.
So what do they do instead?
They ship the instructions, the messenger RNA or mRNA. They send the paper blueprints to the very edge of the growing sheath, and they build the protein right there on site. It's just-in-time manufacturing for a cell.
That is incredibly clever.
It is. But here's the crucial connection. MYO5A, our little truck driver, needs a key to start its engine, and that key is calcium.
Ah, okay, I see the whole chain now. The membrane stretches, the TMEM63A sensor opens, calcium rushes in, the calcium turns the key in the ignition of the MYO5A truck.
And that truck starts walking, driving the blueprints out to the edge. So without the calcium, because the sensor is broken, the trucks just sit there, idle. In the mutant mice, the levels of active MYO5A were down by 42%. So the construction crew is standing at the edge of the wall waiting for the plans, but the delivery guy is asleep at the wheel because he never got the wake-up call from the calcium.
That's a perfect summary. And because the blueprints, specifically for a protein called myelin basic protein, or MBP, never arrive, the inner node just stops growing. That's why the segments are too short. The entire supply chain collapsed.
It's just amazing how mechanical this all is. We're talking about trucks and roads and blueprints and tension sensors. It feels much more like engineering than biology.
Biology is engineering. It's just wet engineering. And sadly, we see exactly what happens when this very specific piece of engineering fails in humans.
Right. The paper mentioned a connection to a human disease, HLD-19.
That's it. Transient infantile hypomyelinating leukodystrophy 19. It's a rare genetic disorder, and it's caused by mutations in this exact gene, TM63A.
And I'm guessing the symptoms look a lot like the clumsy mice.
Very similar, but much more severe in infants. Human babies born with this have serious motor issues. Nystagmus, which is where the eyes shake uncontrollably because the muscles can't stabilize. Ataxia, which is a severe lack of coordination. And if you look at their brains with an MRI as infants, there's a shocking, devastating lack of myelin.
That sounds absolutely terrible, but the name of the disease, transient, that word usually means it goes away.
And this is the paradox of recovery. And honestly, this is the part of the paper that hit me the hardest.
Why is that?
Because the transient part is a lie. Or, at best, it's a dangerous partial truth. As these children grow up and as the mice age, the brain eventually realizes something is profoundly wrong. It switches into a kind of survival mode, a different metabolic program, and it manages to wrap the axons anyway.
So they get better. The MRI starts to look normal.
The MRI looks. Okay, the total volume of white matter, the myelin, it returns to near normal levels. But, and this is the absolute critical point, the geometry never fixes itself. The researchers looked at the adult mice who had recovered. Their internodes were still too short. The gaps, the nodes of Ranvier, were still too wide.
So the wall was eventually built, but it was built shoddily.
Exactly. The quantity recovered, but the quality never did. And the functional result of that? The clumsiness never goes away. The fine motor deficits are permanent.
That is a terrifying distinction. We usually think of recovery in medicine as meaning back to normal, But you're saying the brain just kind of plastered over the cracks, it didn't fix the foundation.
It proves that TMEM63A isn't needed to make myelin. The brain has other ways to do that. TMEM63A is needed to tune it with precision. It's the difference between wearing a burlap sack and wearing a perfectly tailored suit. They both keep you warm, but only one lets you run a marathon.
Let's do one last recap, because that's a powerful point. The link between the calcium signal and the actual building is a motor protein, a little truck called MYO5A.
Right, which is activated by calcium.
And its job is to carry the blueprints, the MB mRNA, to the construction site at the edge of the myelin sheath.
Yep. No calcium, no delivery, no growth.
And in humans, this leads to a disease, HLD19, that looks like a severe lack of myelin in babies. But the word transient is misleading.
Extremely misleading. The amount of myelin eventually recovers to look okay on a scan, but the structure, the geometry, is permanently flawed, which leads to lasting motor control issues.
Wow. Okay, so let's zoom all the way out. We started with a tiny ion channel being physically pulled open. We followed that signal to a calcium spark, which started a molecular truck, which delivered blueprints to build a wall. And we ended up understanding why a child might struggle to walk.
It's a beautiful and tragic and incredibly complex supply chain.
It really makes me rethink how we teach biology. I spent years memorizing receptors and ligands and chemical keys fitting into locks. But this suggests that a huge part of our development is just raw mechanics.
It suggests that physical force is information. The brain is literally feeling its own structure to decide how to build itself. If the cell doesn't feel that stretch, it doesn't build the wall correctly.
Which leads me to a kind of wild thought to wrap this all up. And the paper didn't explicitly say this, of course, but I'm connecting the dots here. If myelin geometry is determined by mechanical stretch during a critical developmental window, does that mean movement itself shapes our brain's wiring?
That is the provocative question, isn't it? If a baby is moving, falling, crawling, constantly stretching those nerves, are they actively mechanically tuning their own myelin?
Like, could physical therapy literally be a way of optimizing the strain gauges in your oligodendrocytes?
It's certainly possible. It implies that stretching your mind might be a lot more literal than we ever thought, that maybe sitting still during development literally leads to generic off-the-rack wiring.
Well, on that note, I think I need to go for a run and tune up my inner nodes.
I think we'll join you.
Thanks for listening to this deep dive. We'll see you next time. Keep stretching.